Good morning, everybody. My name is Camila Abels. I am a member of NSF staff, and I just want to say welcome to our workshop today. And this is the Epigenetics workshop, Reflections on the National Science Foundation's Understanding the Rules of Life program, or we call it UROL. This is a virtual workshop. Could you go to the next slide? This is, next slide. Okay, so this actually first of 4 workshops that we are organizing at the request of the NSF. So this one we're going to speak with the PIs of epigenetics projects, and then we have one on February 22 on microbiome, March 21st synthetic cell, March 28th will be the cost-cutting workshop. Registration and other information about these upcoming events are posted at our project webpage. But if you have any questions about these upcoming events, you can contact Crystal Saunders. Her email is up there. And also, I just want to remind people that this workshop will be recorded, and materials presented on the slides, verbal comments, and comments submitted via Slido may be used in the workshop proceedings with attribution. And now I turn it over to Connie.
Okay, could I have the next slide, please? Okay, so I'm Connie Mulligan. I am a member of the workshop planning committee. All the members are listed on the left side of the slide there, and some of the committee members are present here on Zoom. We're all researchers and academics who were requested by the NASEM staff, who are listed on the right-hand side of the slide, to participate in this NSF requested review of the UROL program. And so this review is happening through the series of 4 workshops that Camilla talked about in the previous slide. And as she said, the epigenetics one is the first of the workshops. And so today at our workshop, we're also going to have Carol Burkhauer, who is a science writer. So she will be taking notes throughout the entire workshop and then write a report covering everything we discuss, and particularly, our consensus on the achievements of the UROL funded projects. Could I have the next slide, please? So today we have 3 moderators. I'm the moderator from the planning committee, and I'm joined by Sasha Gimmelbrandt and Kunal Rai, who were asked to participate specifically in this workshop in order to broaden the epigenetics expertise that we have here as we address the different questions we're going to discuss today. And then for our discussants, we have 6 PIs of currently funded UROL epigenetics proposals who are going to, guide our discussion as we, cover the different achievements and challenges of the currently funded projects. And if anybody's interested, all the bios for us moderators and the discussants and the funded projects can be seen at the link on the bottom of the slide there. Next slide, please. So I'll go over the agenda for today. So we're in the middle of welcome and introductions, and then I'm going to cover the goals we have for today's workshop, give a brief overview of NSF's UROL program, and then finish up with our specific expectations for the workshop. And then Kunal's going to take over, and he's going to present a summary of the responses from the PIs of the UROL epigenetics funded projects that they sent in response to questions that NASEM staff sent them. So we have, like I said, we have 6 currently funded PIs as discussants here. There were responses sent from slightly more PIs. It's all anonymized on the slides, but then the 6 PI/discussants, will chime in to, give more, fuller responses to those questions during the discussion section. And so that's what's going to come after the summary. We're going to start a discussion where everyone can participate, moderators, discussants, the audience. Kunal is going to start that off because we have the discussion section divided into 3 sections. The first part that Kunal is going to start with will focus on scientific achievements thus far from the funded projects, as well as rules of life that are beginning to emerge or have been identified or can be tested. Then Sasha's going to take over for the second part of the discussion and focus on multidisciplinary research or the multi-team approach and discuss achievements. So, things that were achieved through a multidisciplinary approach that would not have been possible if the research was situated in a single discipline. Then also challenges to taking a multidisciplinary approach and different aspects of education and training and outreach that also emerge from the multidisciplinary approach. And then I will follow up with a discussion on societal issues that have been addressed by the proposals, and then future questions to be addressed in future iterations of proposals. Then we're going to take a brief break when the moderators and NASEM staff will scramble to synthesize some of the big ideas that emerged during the interactive discussion, so that then when we come back after the break, Sasha's going to present the attendees' views on common themes and try to get a consensus on, the most important ideas to emerge from the workshop today. And then I will finish up with a quick summary of the key outcomes and themes. So that's what we're going to do today. I don't think it's going to take 40 minutes for us to cover all the way that first section all the way through workshop expectations. So we should have more time for some of those other sections of the workshop today. So could I have the next slide, please? So the goals for today's workshop: we first want to discuss any scientific advancements that have been made thus far from the funded epigenetics projects. And we realize that it's still early days. A lot of projects have just started. Very few have actually completed. So we acknowledge that it's early days to be talking about the achievements that have come about so far. But NSF wanted, to do a review at this stage of things just to see how things are going. So what we want to do is ask, the PIs, the discussants, to talk about what they've learned so far from their research projects, and also move to a broader perspective to think about what rules of life they're starting to hone in on. So a key part of the project is this multidisciplinary approach. So we want to discuss how any achievements or contributions to the research have been informed by the multidisciplinary approach. So that's the second key part that we're going to look at: how the different research groups incorporated a multidisciplinary systems level approach into their project, and then really dig into what was accomplished, what were the challenges, have they been addressed, are they being addressed, what could we think about for future iterations of proposals to better enable multidisciplinary approaches. And the whole idea behind this really strong push of a multidisciplinary approach is basically that we have a better understanding if we can integrate expertise and people and concepts across different disciplines. And so this whole idea of team science is a topic of research in itself. And that's what the final workshop is going to be on, that Camilla mentioned, the cross-cutting workshop. But so here we are going to talk about, how this multidisciplinary approach contributed to any achievements, and also looking to the future, how a multidisciplinary approach can help, ground research to look at even more complex, even more multi-systems level questions. And then the third focus of the discussions, and then sort of our takeaway message from today's workshop, is going to be identifying societal issues and needs that are being addressed by the currently funded projects, future societal needs that have emerged, as well as future follow-up scientific questions that can be addressed and are grounded in, the research and achievements that are currently emerging. And then finally circle back to, what really is the main focus of the Understanding Rules of Life program, which is to really identify rules by which biological systems are governed that are generalizable across fields and scales. Next slide, please. So the URAL program emerged from NSF's Big Ideas program, which was just basically to identify big, bold ideas about how life works, how biological systems function, that would cross-cut disciplines, would cross-cut directorates, would require the integration of different skills and concepts and theories so that we could get big, bold answers to these questions about how life functions. Then the Understanding Rules of Life was one of those ideas that emerged from the Big Ideas project, and it was based on developing a predictive understanding of how key properties of living systems emerge from the interactions of functions such as genomes, phenotypes, and dynamic environments. So then getting at this rule, so the whole idea is to identify rules that function at multiple levels by addressing, specific fundamental questions in life sciences that cross different scales, and then produce rules or broadly generalizable results that will allow us to predict changes in these biological systems. Next slide, please. So the Understanding Rules of Life program has been very successful, almost $100 million in grants awarded as of the end of last year, 63 different biological systems being studied, and now flipping down to the bottom right corner, so 79 different institutions have received funding, 6 of which are minority serving institutions represented by their PIs. And 35 states have been represented by PIs or co-PIs on the URAL funded projects, and 60 projects have started as of the end of last year, 21 of which have been completed or are in their final year. Next slide, please. So the key goals of the URAL program are to, first off, enable discoveries that will allow us to understand interactions between different systems that will then enable us to identify causal and predictive relationships across these systems. In other words, the so-called rules by which biological systems function. And then having identified those rules, having done this sort of science will give us the capacity to approach ever more complex questions. Thirdly, will allow us to train the next generation of researchers in team science, in multidisciplinary research, so that they are, so that's the way they think, essentially, that their thinking and the way they develop research projects isn't bounded by any one single discipline, but that they more naturally, more organically think across disciplines and can, develop research that gets at these big ideas like understanding the rules of life. And then finally, to foster more research that is collaborative and convergent and multidisciplinary, to consider multiple levels of organization and complexity as we address key questions in the life sciences. Next slide, please. So there were 2 calls for proposals, for URAL proposals focused on epigenetics, both in 2019. And those proposals were required to integrate perspectives and research approaches from more than one research discipline. And this interdisciplinary scope was also required to provide training and outreach possibilities to train the next generation of scientists in a diversity of approaches and to engage society more general. There were two types of epigenetic projects. The first one had a total budget of $500,000 spent over 3 years, and the second one, a total budget of $3 million spent over 5 years. And the next slide, please. So to follow up, the specific goals, expectations for the workshop today are that we're going to address these 3 points. First of all, the scientific advances and rules of life that have emerged or are emerging from the funded projects. Then secondly, really dig in deep to this idea of multidisciplinary research and multi-team research, the achievements, challenges, and education and training opportunities. And then finally, to look at societal implications or needs that have been addressed and can be addressed, and to identify future scientific questions that have emerged from the research. And throughout all 3 of these points, we want to be thinking and discussing about suggestions for improvements for future iterations of the URAL program. And so just to reiterate what Camilla had said, we've got the committee members, the planning committee members, the NASEM staff, and the discussants all attending through Zoom, and Zoom participants should use the raise hand feature. And then we've got attendees watching the webcast who are going to use Slido. And then the next slide gives some information on how to do Slido. And the NASEM staff is going to be monitoring those questions and then feeding them to us as moderators to go ahead and throw them out to the group for the workshop. And so with that, does anybody have any questions before we move on to the actual, scheduled stuff? Okay, with that then, the next slide, Kunal will take over and summarize the PI responses to the questions the NASEM staff sent. Thank you.
Hey everyone, my name is Kunal Rai and I'm at MD Anderson Cancer Center studying cancer epigenomics. My job here is to read 20 slides or so. So please bear with me, you'll hear a lot of my voice here. And we would probably be using this as a platform for the interaction session, right? I'll be reading a lot and trying to see if I can summarize some of these. But these are your words, the PI's words typically, and I'm gonna try to do justice to accurately saying what you guys have written. So please bear with me. And if since this is meant to be eventually more interactive, so if you think I'm saying something wrong that you wrote here, please do correct me, stop correcting. This can be more informal than just me reading things. So, and we will obviously be bringing this up back. It's just the way the agenda is organized right now. Okay. So the first couple of slides talk about scientific advancement that the PIs of the projects provided, whoever responded to the survey, of course. So the first point here is that the PIs have determined that plants have a heritable growth in response to high CO2, and there is more of this later on, so we can discuss that. That's in one line the achievement so far from the PI. The second point here relates to probably a modeling framework where PIs are using this as a quantitative tool to study biological systems in which the epigenetic state is regulated by specific non-coding RNAs, and that's laying a foundation for how the non-coding RNAs might be leading to transcriptional silencing. Again, I'm not entirely aware of your project, so we will be discussing these, and at the time of interaction, you guys can come up and please tell us more about it. We can all have a fun, more academic discussion rather than just reading stuff here. The third PI has learned that bumblebees respond pretty quickly to the cold shock at the molecular level, and they utilize potentially DNA methylation changes for that, and that occurs in the regions where there is a lot more nucleotide variability or diversity. Another PI's work has revealed how epigenetic state of corals depend on their environmental history, and how maternally experienced conditions influence provisioning to or methylation state of the offsprings. So now their work is providing new tools for predicting equilibrium states in systems that exhibit hysteresis, and describe how the interaction of coral sim points can modulate coral bleaching responses. The next PI is looking at how how the adaptive processes might look like when rapid climate changes might occur in a single cell or in multicellular organism. And we would love to hear more about this afterwards. And finally, in this slide, at least, there is one more. And the next PI has evidence that epitranscriptomics modifications of host and viruses can both be modulated by the microbes, and that's a very interesting, I think, observation where an external agent can utilize epitranscriptomic information for and change the epitranscriptome of both host and host viruses. But would love to hear more as we go forward. Next slide, please. The next PI has been studying the YTH domains and computationally predicting what kind of reader domain this might be. And I think their conclusion, or at least computational prediction, is that the YTH readers can recognize a diversity of modifications with different strength, and this is likely not specific to one kind of M6A modification, for example. But again, we can discuss this afterwards. That's what I read into that. The next PI is testing relationships between energy metabolism and epigenetic control of gene expression, and underlying change of genome function to generate how the phenotypes are generated eventually. And they're specifically characterizing how symbiotic nutrition interactions and metabolic states modulate epigenetic dynamics, providing intragenerational and transgenerational memory. And we all know that's a very important epigenetic question as such. And that eventually leads to obviously how organisms work, and how the evolution eventually works. And for the next PI, who lists mostly the deliverable at the end of the project, they're trying to build wet lab and computational pipelines for nanopore-based long-read sequencing based approaches for epitranscriptomic datasets. So that's obviously very important for getting to the next level of understanding in epigenetics as to these long-read sequencing-based methods. And lots of people are actually attempting this, so that's obviously good to see here. And finally, the last PI says that the major achievements are associated with writing, testing, analyzing, and improving the computational setups for simulations and free energy calculations. Next slide, please. Next slide, please. Yeah, okay. The rules of life slide, yeah. Next one. Thank you. Okay, so we next asked to the PIs what are the rules of life they've learned, and eventually this is what the discussion is going to be based on. But this is like same PIs, obviously building on their scientific advancement and trying to project what are the rules of life mostly here. And the first one relates to how the increased rate of growth they see in high CO2 condition is actually a heritable condition, and that obviously speaks to epigenetic features here. And what this might suggest is that in conditions where if you have high CO2 rise, and this could probably broadly apply to other conditions where we see environmental changes, and where the epigenetic, probably rapid epigenetic transitions happen, and they are adaptable eventually, to the rate I guess I didn't think about. I didn't imagine that to be. So again, love to hear more later. But this suggests that the biomes such as forests and grassland, which are built of plants, where as CO2 rises globally, these plants adjust pretty quickly to the growth and stress, and they probably utilize DNA methylation based epigenetic information, I guess. The next one is that the next PI is learning rules of interactions and regulations among different layers of epigenetic regulation. So there's not much detail here, so we'd love to hear more about what kind of epigenetic regulations, etc., during the discussion session. And the next PI has learned that, and this is important, that metabolite production occurs at the same time the transcriptional changes occurs in bees during cold shock, which means that they are not necessarily, I mean, which may mean they're sequential process, but it has to be pretty rapid, or they may be interconnected so that they occur pretty simultaneously. And after that, the next one is how DNA and RNA are modified in eukaryotic cells and the consequence of cell biology. So again, this is more generic and would love to hear more, but they're probably identifying rules that may be governed. And I think this is more related to transcriptomic based learning. Finally, the next PI states that obviously this is premature, and that makes sense. We've all gone through a COVID period with minimal progress, and saying rules may be too premature. So they are learning the rules that govern selection of phenotypic plasticity via epigenetic mechanisms. They are describing the constraints of this plasticity, determining the mechanisms of these epigenetic modifications, and rules for resource exchange in mutualisms. And I'd love to learn more about that, what that means. But basically, this is obviously the whole field, and many PIs study this. But so this is again generic. But love to hear a little more details during the interaction session for some of these. Next slide, please. Yeah, this is the next point comes from from the PI who studies YTH domains, I guess, and so there's different modifications can be read by same binding pocket. So that obviously has implications in potentially in epitranscriptome relaying the information to the next level, and potentially in drug development to such pockets. They have observed that this overall aromatic mature nature of binding core is present in many species, and these domains are overall discriminating for the RNA modifications that specifically involve methylations, but just not M6A. Okay, next point, next PI responded with who are working in coral epigenome, that they're seeing the same epigenetic mechanisms being present across all species, and they believe, and this relates to DNA methylation information in gene body, etc. And they state that this is in contrast to the epigenetic patterns of many vertebrates, where potentially slightly different methylation information is utilized across vertebrates, which makes sense. I mean, the yeast to the Drosophila to humans utilize potentially different DNA methylation patterns, and some of them don't even contain DNA methylation. So I would love to hear more. Finally, the last point here is the two key roles of light that were learned was chromatin, chromatin is just not a binding pocket, but in fact, it's an active catalyst that promotes complex formation. And I really like that point personally. And finally, stress is not necessarily detrimental to cells, but is key determinant for epigenetic adaptation. And that's something I think we need to think a lot more about as our world gets into more stressed environment, of course. So how that it will lead to adaptations for future generations. So of course that's critical. That's critical piece of work, I guess. Next slide, please. All right, we next asked, while the UROL program asked everyone to work in these team efforts, obviously bringing people from PIs from different disciplinary teams to work together. What were the practices that were done, and what were the challenges that they came across, and what were their learnings? So this slide speaks to the practices. Of course, during Zoom, we all learned to be more savvy with all the computer things, all the electronic media and various apps, etc. So Zoom, Slack, I mean, is probably become way of life for most of us. I mean, and that was obvious that we would see here. Uh, what were the practices of mutual trust, commitment, and supporting each other, and desire to build strong connections over different research areas? So that's obviously key, fundamental to multi team research, and that's again good to see here. The other practices for other group was that their proximity helped in their communication, and obviously they also utilized the resources and the cloud and sub meetings, and their leader was pretty good at setting up goals, timelines, and responding to people's needs. So that led to possibly uh, good progress in the project. The next point speaks to what were the practices on the practices side. Says field specific perspectives were utilized, I guess, and modeling sizes, for example, familiar with the species studies, stuff cells. PI has learned a lot of them, but also from the genomic side, there is a lot to learn about the scale of G cells and genes. So what I'm reading is essentially the PIs with different expertise came together and try to learn each other's relative areas of expertise and work together to sort of go on the same page. Next slide, please. This is again other more responses on the same area of practices. Those are regular communication, and that obviously helps, and probably the P and the group members had long-term relationship among themselves, which led to easier communication and probably success in working together. And next point speaks to how it is important for PIs to like each other, and of course, none of us can work without that. So, and a shared sense of scientific mission, and that's likely why you came together to write a UROL proposal that was funded with overlapping interests. And they met monthly, or they still meet monthly to ensure project integration, Zoom, etc. Of course, same things. The next point speaks to how that group has taken systems level approaches for multiple aspects, and they're trying to study epigenetic mechanisms and organismal and ecological performance, and they built this dynamic energy budget model. And again, would love to hear more on this. DB theory is based on metabolic organization and dynamic representations of the flow of energy and elemental matter within an organism. So would love to hear more. I'm not familiar with this. And finally, these next group combined the in silico approaches with the experimental biochemical techniques, and these different groups of people came together to understand the question they were asking. So next, this is good to see that these multidisciplinary teams utilize these practices of regular communication, utilizing the tools, resources they had available with mutual trust and common scientific goal to come up and deliver on these projects. So obviously that suggests that some of the URAL objectives were met through these multi PI collaborations within these teams. Next point, please. Next slide. Okay, what are the challenges? So the first point on this speaks to the fact that when you work with standard large data sets on, I think, organisms that are utilized regularly, for example, humans, mice, etc., this current set of pipelines and standards and softwares work well, but they had to probably they had challenges to build customized pipelines and couldn't use the standard ones for their species specific interests. And they had to do more thinking on being explicit about assumptions that were brought to the data collection, designing experiments, and having conditions. So that's good. I mean, that's what we wanted you guys to do. I guess the URAL program wanted you guys to do is to think more, come up with novel thinking, even not just using standard sort of pipelines. And this PI states that it would be nice to receive the money and large sum at the beginning rather than spread out over 5 years. And this is one of the points for discussion later on. The next point speaks about how they had obviously many challenges through COVID, and that is a common theme, which we've all been affected by. So nothing we can do about that. But they had to then work through COVID and try to go in their different directions. So that might be a challenge. Rather than doing a team science, they might be doing individual science. Next point is on keeping all the PIs on the same page on processing samples, because of COVID mediated delays in attendance and joint meetings, etc., were not done. PIs were changing institutions, and that led to various issues, personnel and budgetary issues, and also they had problems with sharing data across institutions, which obviously can be a challenge in some. I think all of us have encountered, especially if they are from humans, where the privacy issues come into play. So we can discuss these during the discussion point. The next point speaks to how the money was distributed, and budget was provided separately to each co-PI and collaborators. That created difficulty for the lead PI, where they were not able to manage PIs who were not performing. So that would be something, I think that would be a good point for NSF to think about for the next grant. And the next point speaks to they would want to have a workshop of common experience among grants awardees, something I think we are having now, and would be good so people can share solutions they may have found. And I think that's a good idea. Having these intermittently organized would be a good idea for people to learn from each other. And I guess the next point, and the point that this PI is making, is about how they had to motivate their graduate students for these different challenges they were thrown at. So I guess that point comes up again in a later slide. Next point, please. Next slide, sorry. Yeah, the next challenge is because the projects require these multidisciplinary approaches and they utilize multiple labs, I guess synchronization takes time. And yeah, indeed, they present unique challenges from science, as well as cultivating a shared vocabulary. And somebody else later on, the last point here, talks about translator scientists, which I actually loved. So having subgroups of folks. So I'll just jump to the last point here, which talks about how they solved that sort of problem, for example. So these last point, PI started a team of people with somewhat overlapping experience, and they formed subgroups that met individually and then came together to report the integration discussion. And they served as translational scientists that probably worked across to for example, telling the biology to the computational biologists, etc., and vice versa, and so on. So I think that's a good idea in any of the multidisciplinary teams to have these common people who have sort of shared expertise, or probably jack of all, and to really make the other folks feel and make the project understand better. The second point on the slide is more on they could benefit more from machine learning and artificial intelligence approaches to examine the seemingly endless number of interactions between epigenetics, environment, gene expression, and their feedbacks. And I think these kind of approaches are being utilized now. I'd read the point, given that the current working hypothesis on epigenetic state linkage to gene expression regulation only explains a fraction of the variance, it would be beneficial to take some non-targeted approaches to data integration in addition to our targeted hypothesis on specific insights. And I think that's a very key important point scientifically. This probably doesn't speak to this overall theme of the multidisciplinary research, but I think it's important for us as epigeneticists to think differently on how the role of these epigenetic events is only to modulate gene expression. There is likely much more than that, and that needs to be understood through these novel AI-based approaches that are completely unbiased. So completely agree to that point. The third point speaks to how this PIs had how this group had computer scientists, mathematicians, bioinformaticians, and plant phenotyping labs, and how they came together. It was obviously difficult to get the computational scientists and mathematicians to understand biology, something in my lab we deal with every single day. Yes, but it is what the UROL program is about, and that's a great strength here. Otherwise, you might as well be doing an R1. Okay, and next slide, please. Yep. Next, so this talks again about similar aspects. Again, quantitative sciences and biologists. There is a barrier, but we need to, as a society, as epigeneticists, as a group of folks, we need to overcome that, because that's where we're going to make next set of big advances, or the rules of life that we speak about here. Next point speaks about how the pandemic had poses challenge, but they also talk about how it is critical to support the trainees during this period, and that would to help overcome this. They also talk about how PI moving to another university that led to rebuilding and delays. Makes sense. Next point talks about the reconciling the in vitro and in vivo behavior, and so we can talk more about that during discussion. Again, the next point is the same that we've been bringing up: a common language for integration from scales to empirical to theoretical. I mean, this goes one level above. And yeah, we just talked about this, how taking approach of overlapping people with overlapping expertise would be good to have. And the final point on the slide is parameterization aspect. It always evolves with new techniques or methods emerging, and understanding how models are different or good for prediction is not always obvious for the experimentalists. And a lot of times, I think even computational biologists or mathematicians rely on experimentalists to show or prove so that they can modify their model eventually. So this is, I think, a continuous flow between the two disciplines. Next point, please. Um, okay, challenges. So starting from realistic grounds to produce quicker results. More in-person meetings would be better now that you guys we've come out of pandemic. I think this would help. This program would benefit from facilitated interactions of the funded teams to really generate the rules of life. And that I think I hope NSF will keep in mind. I think there needs to be more interaction between the different teams. Keeping every different lab focused on the work was one of the challenges. Uh, COVID, yeah, we talked about this, people changing institutions, etc. And the next challenge is some labs were spending money from the project, focus on their main research interest, but not producing results for this project. And I think this is part of the point. This is part of the job of the lead PI to keep everyone, focused on the project. So I guess we can discuss in the discussion section how to sort of manage that for people who are experienced in leading these multi PI teams. And we can understand. I mean, this leads to sort of friction, and as you reach towards the later year funding, you haven't finished things. So okay. Uh, Again, I think the same point is coming again. That workshop to share common experience among grant awardees would be a good. And yeah. Next slide. All right. Now it moves on to education and training of the grad students and postdocs, etc. So obviously the grant provided training opportunities for these folks. But the greatest value came from researchers entering a multi-institutional network, and that's good to hear that people made more contacts and probably was beneficial to everyone. The students were forced to confront research outside of their comfort zone, and this is definitely good. You guys can read the things that I'm not reading if you're more interested. Of course, I'm just trying to fill the more main points. Uh, and again, the next point is similar in that students across institutions and disciplines, and that requires coordination, frequent meetings. I think, but it is for good for students in the larger run, and this is where the world is going, and we should all think about that. Students need to learn the language from others, absolutely. Um, yeah, same point. And finally, students by engaging, mentoring the students by engaging them through active learning and critical thinking beyond learning the facts. I think that's a general, I guess, struggle for many of the PIs, and who can do the well are really great mentors, and end up training really great scientists. So that's part of what we're trying to do here. You're gaining additional experience, mentoring additional students. Yeah, it's good. And again, it helps all of us as mentors for whatever we do. And it's good to hear that you all is helping you guys all learn, become better mentors for your students. That's good. Next slide. I think that, and this is an important piece on top. The first point there, we had the PIs had to educate their trainees uh, to actually present the research in a broadly understandable way, and that will again go a long way in helping them become better scientists or better communicators. And uh, COVID Zoom life for this. I mean, the better part of the good side of COVID was that we all learned to do Zoom, and we were able to reach out to colleagues who are across the world, not just within different positions in US. So that was also good. And that's good to see here. Uh, I think the other points are similar in nature, so I'll pass on. Next slide, please. Yeah, I think these again, this is about societal issues or scientific questions that were addressed, and I think the responses are similar in a lot of ways. So. Uh, I guess you guys can read this. We can give you a few seconds, but it's all about similar findings as previously, because this is all about scientific questions. I mean, foundation to understand the mechanisms of transcription science by non-coding RNAs. We covered that during the rules of life, or the other group mapped out patterns of DNA methylation across genome. That's good. This has obviously, as a society, helped us learn from uh, different parts of life, whether it was corals, whether it was bees, whether it is some more biochemical approaches, whether it's humans. So I think what we are learning together with this program, I guess, is a lot more. Um, can be. I think it's at least somewhat partially um, fulfilling the original challenge that was put on through these different different groups that you guys are making great progress, I guess. Yeah. I again, these are same points as previous, so I'm not gonna belabor upon this. Next slide, please. And Connie will talk about this later during the in discussion too, or just will take on from here. Next slide. This is almost societal issues. So, Connie, you want me to read this, or you want to take this on during your? I guess one of the key
Yeah,
things
yeah.
is how.
This is largely duplicated in my slide for the interactive discussion. So,
Okay.
we could possibly wait. And I think this is the last slide for the summary
Right.
of PI responses, right?
Yeah, and then yeah, exactly. So I just start the discussion on the next slide with some of the points from rules of life. So
Right, but I thought maybe before we get into the discussion, so that we can kind of get everybody used to talking, maybe we could first have the moderators just give a brief, description of our research or expertise, and then go through the discussants. Just take a couple minutes total to get everybody talking. So I'll just go ahead and start. Again, I'm Connie Mulligan. I'm at the University of Florida. I'm in the Department of Anthropology and also the Genetics Institute, and I study I look for possible epigenetic mechanisms to mediate the impact of psychosocial stress on different health outcomes. And so my research is very multidisciplinary, because I work with cultural anthropologists and clinicians and heart researchers. And, my training is in hard sciences, but now I'm in a social science. So I'm very interested in what works and what is more challenging about multidisciplinary approaches. So now maybe Kunal and then Sasha, and then we'll move to the 6 discussants.
yeah. So sorry, you had to bear with me for last 20-30 minutes reading these slides. As I mentioned earlier, I'm Kunal Rai. I'm an associate professor of genomic medicine at MD Anderson Cancer Center. I also serve as scientific director of an epigenomics therapy initiative at MD Anderson, which is geared towards clinical translation of epigenetic therapies and novel epigenome targeting modalities. My research focuses a lot on epigenome. We try to understand epigenome in cancer and different aspects. And we utilize high throughput methodologies, cutting edge single cell and bulk level methodologies. And that includes wet lab science as well as computational science. So half my lab is computational, half my lab is wet lab. And then we also have various approaches. We use patient samples, of course, plus but cell lines, and then mouse models and zebrafish models, etc. So we learn across various species. So becomes quite interesting and challenging
Okay.
to do all of
Sasha?
that. Yes. Sorry. Yeah. So that's pretty much in lots of multidisciplinary work in my own lab as well. So love to discuss more from participants and learn what their experience was here.
Hi, I'm Sasha Gimmelbrandt. I work at Altius Institute for the past couple of years, and I moved my lab from Dana-Farber, and I have long been interested in sort of epigenomic mechanisms that control variation between individuals and between cells. Like Kunal, I have long been sitting on two chairs between, I was trained primarily as a wet lab biologist, and for the past many years, I've been sort of combining computational methods with wet lab experimentation. So most recently, we have been very interested in long-read sequencing and its use for understanding how things are working in the epigenome. And that's what my background is, and I'm very much looking forward to hearing more from the PIs.
Okay, so maybe now we could hear from the discussants. I'm just going to go in the order that you guys, it's an alphabetical order listed on the slide. So Lydia Contreras, could you talk first?
Yeah, sure. Good morning, everyone. This was a great introduction, got a lot of thoughts going. So I am at the University of Texas in Austin, and our lab is a very interdisciplinary group that takes an experimental molecular approach to questions about how the epigenome makes an impact to environmental stress responses. We are specifically interested in RNA molecular interactions between RNA and proteins. And we have a lens for that from the RNA disciplines, biochemically RNA disciplines, but also from design, largely quantitative backgrounds.
Okay, thank you. And Julie Hotop?
Hi, I'm Julie Dunning-Hotop. I am at the University of Maryland, Baltimore School of Medicine in the Department of Microbiology and Immunology and the Institute for Genome Sciences. I have a wet lab and a computational group, kind of bridging the two, and focus on the genomics of host microbe interaction, kind of broadly, with a specific emphasis on Wolbachia host interactions. And so my collaborators are a virologist, a biologist who is also interested in host microbe interactions and symbiosis. And then me, and we are looking at epitranscriptomics in a multi-organismal system.
Thank you. Jeffrey Lozier?
Hi, I'm Jeff Lozier. I'm a professor at the Department of Biological Sciences at the University of Alabama. My lab is sort of also, like everybody else, multidisciplinary. We do sort of molecular ecology of a wide range of organisms, from freshwater mussels to crayfish. The bread and butter organism that you may have seen on a few things is bumblebees. And we're particularly interested in phenotypic variation in bumblebees that live in sort of complex heterogeneous landscapes and how that drives phenotypic shifts. We've been focusing recently on sort of the molecular evolution and local adaptation associated with elevation, and in particular with cold response, because bumblebees are very cold adapted organisms. So we've been transitioning into understanding both the physiological and molecular mechanisms involving sort of cold response in bumblebees. And we had previously identified variation across elevations in the response and the phenotype, and that this maybe was associated with transcriptional variation. And so we don't see a ton of nucleotide differences. So we were wondering if perhaps epigenetics was playing a role. And so that was what led to the project, and I feel fortunate that we to have data at the right time and place for this grant proposal.
That sounds great. Thank you. Holly Putnam?
Hi, I'm Holly Putnam. I'm an associate professor in biological sciences at the University of Rhode Island. And we have a multidisciplinary team working on this project. We're focused primarily on an organism that's a holobiont, symbiotic reef-building corals, and the connections of the different partners, and really interested in how corals in particular, in this case as our non-model model species, respond to environmental change, and what are the underlying mechanisms by which corals and other organisms transduce the environmental signals into the plasticity that we see and in particular with the coral reef crisis and applicability to societal outcomes in thinking about what role will epigenetics play in sort of buffering the response, generating phenotypic plasticity, and leading to potentially positive outcomes for corals, both within generations and across generations.
Thank you. Kaushik Raghunathan?
Hi, my name is Kaushik Raghunathan. I'm an assistant professor in the Department of Biology at Brandeis University. My lab uses Fission-East Schizosaccharomyces pombe as a model system. And we've been using the system in order to study mechanisms by which epigenetic systems are misregulated. But the misregulation process is very useful because it enables adaptation to be possible. My lab does a bunch of different things that includes both biochemistry and genetics in this organism. The UROL team that I'm a part of and that I lead involves biophysicists, computational biologists, and synthetic biologists. So it's quite an interdisciplinary team that I think meets the needs of this highly interactive project. And thank you again to everyone for organizing this event.
Thank you. And Keith Slotkin.
Hello, my name is Keith Slotkin. I'm a principal investigator at the Danforth Plant Science Center in St. Louis. I'm also a faculty member at University of Missouri. My lab studies the initiation of epigenetic silencing on transposable elements and transgenes in plants. The UROL team that I lead is investigating the plant response to high carbon dioxide level, both phenotypic response, so we image plants hourly, as well as molecular and epigenetic responses. Primarily, we're looking at ATAC-seq and Hi-C and RNA-seq. And then we work with computer scientists and mathematicians to try to combine that data and look at how those phenotypes persist even into the next generations after those plants are not grown at high carbon dioxide levels anymore.
Okay, great. So that's all the moderators and discussants. So now, Kunal, do you want to start us off on the interactive discussion?
Sure, I just put up some points from the other, like the first few slides here. And again, since you guys are this is your work and research, would be good to hear from you guys, I guess, and would be for us, all of us together to think about how to really, I guess, clearly spell out the rules of life here, right? That you guys are learning. And so I guess we could start off with the first one as to how the plants adapt to this high CO2 environment that could potentially be important in a societal way when we have these high climate issues that we are having or predicted to have as we go forward, as to how our learnings from the epigenetic front could be utilized or just in general, I guess.
Yeah.
Sorry, if I could just sorry, I didn't mean to interrupt again. But again, everybody should feel free to chime in, to expand on any of your responses, that Kunal presented, but, give more than what you were able to put in your short responses. And also the idea is that, by being in this workshop and thinking about what the other labs are doing, that it spurs you to think even more broadly about your research you're currently conducting, any ideas you have. I mean, it's intended to be kind of a brainstorming session. So move outside of your area of expertise or even comfort zone to think big about what, the rules of life might be. And that's the last time I'll interrupt.
And how NSF should keep funding you more.
Exactly.
Okay, I think that's the first project here is the one that I lead, and what we found is that, like other stresses, plants respond to stress, but then often set up gene expression programs epigenetically that persist into the next generations. And the same is true for carbon dioxide. If you grow most plants in carbon dioxide, what are called C3 photosynthetic plants, which is about 85% of all plant species, crop, forest, grassland, whatever, they will grow at an accelerated rate in high carbon dioxide because they're using that as a general input for sugar production through photosynthesis. And then if you take them away from that high carbon dioxide in the next generations, they'll continue that accelerated pace of metabolism that was set up originally at the high carbon dioxide level. So we've studied this across a number of different plant species and found that generally many, or I should say all C3 plant species that we've tested do this, including really representatives of early plant lineages like the moss Fiscometridum, which is just a common moss that people grow in the lab. So this is a representative of early plants. So we've been exploring molecularly and trying to find, which genes, what molecular mechanisms. We now understand. Our project is now into the 4th year, so we now understand what DNA methylation specific pathways and regions of the genomes are responsible for that. So DNA methylation, if we use DNA methylation mutants, that original response to high carbon dioxide is there, but then it fails to persist in the next generations. So we know DNA methylation is really important, and we are trying to then interconnect the plant's form and 3D organization with the genome's 3D organization through Hi-C. And that's really where we need a lot of those computational biologists, because there's this grand challenge of kind of connecting phenotype to genotype, and particularly in these different data sets that goes beyond, what I can do as a trained bench scientist into the world of like AI and computer science and mathematic modeling.
That's a great point, Keith. I mean, I just want to ask you one, how long do you see these effects persist after you take them out of the high CO2 environment, for example?
Yeah, it's strongest in the first generation. So the seed produced at high CO2 into the next generation. It can persist into 1 or 2 more generations, but it's at a diminishing amount. So our project really studies that first generation, trying to identify and dissect the mechanism that's responsible for that propagation of that state that was set up in the original generation grown at high CO2.
So I have a question. So the ability of the plants to maintain that accelerated growth into future generations where there's not an increase in CO2, do you view that as adaptive, that it's somehow good for the plant long term? Is it maladaptive, that the plant can't adapt, that quickly to the new environment? Or is it just neither? Is it just the consequences of the way in which that increased growth is regulated?
No, we think of this as adaptive. There's a well-described phenomenon of priming in plants. And what priming means is that you're going to give a plant a stress that it's going to overcome, let's say heat stress. And then later in the life of that same plant, it is better able to deal with when it sees that stress again, even if the stress is more intense than plants that were never stressed in the first place. So it's essentially a cellular memory that, okay, the heat stress may be coming. And agriculturally, this is used on a grand scale where companies that sell seeds to farmers will purposely stress the generation before that, so those crops that the seeds that they sell will turn into crops that are more stress resilient than an unstressed plant might be. So we think this is kind of a cellular memory of watching out for what may be coming that either the parent plant or the plant in the same generation has already experienced.
Okay, thank you.
Wonderful. Holly?
Yeah, I wanted to follow up, and you mentioned DNA methylation and DNA methylation mutants. And my question is, you also mentioned AI and things going way beyond probably single genes. So I just kind of wanted to get a sense in your system of what your thought is on the regulatory role of DNA methylation relative to the phenomena that you're seeing of this enhanced growth in terms of global expression linkages or really targeted sets of genes or individual genes, and when you think sort of you might go with certain approaches that are, knock everything out versus these are a few key genes that we think the regulatory role is in.
Right. So targeted is the answer to the first question. So this gets right into my comfort area because I've studied DNA methylation pathways in plants for a long time. And so we have different mutants in the model plant Arabidopsis thaliana that we've used in this experiment that can break apart maintenance methylation at CG nucleotides or maintenance methylation at CHG, where H is any nucleotide but G. So there's any cytosine in a plant could be methylated, not just CG. And then we have other mutants that can maintain methylation fine, but can't add new methylation. So they're mutated in what we call de novo methylation, which is a separate pathway than the maintenance of that methylation. So we've been able to test all of these. And so we understand which de novo pathway is important now for establishing that DNA methylation, and then what maintenance pathway, once it's established, is important for maintaining it. And you need both that initiation and maintenance to propagate the responsiveness to CO2 to the next generation. So this has been the success so far, and we have actually a paper under review right now that details these mechanisms. Now, as far as where in the genome that's happening, that methylation, we can identify differentially methylated regions. And we can see that there is an enrichment of phosphatases, phospholipases that were near these differentially methylated regions. And that makes sense because other stresses and the propagation of those stresses from generation to generation have found these same kind of gene sets. So we have yet to identify the gene, and it might not be a single gene, but we're at least down to kind of a candidate family that have popped up in a number of analyses, both in my group in this UROL project, as well as in the literature, that seem to be playing a major role in setting up these rates of and sensitivity to stress in the next generation.
Thank you.
Yep.
Lydia, you want to go next?
I'm sorry, I'm not hearing. You might be muted.
So sorry. Yeah, I was saying I find that whole DNA methylation and the persistence pretty fascinating, especially coming from the lens of RNA-based epitranscriptome responses, where the whole question about how long the responses persist and can you predict that and how is that related to any molecular patterns is still very vague. So as you were describing your project, Heath, I'm just wondering, and you kind of started talking about it at the end, when you look at a DNA methylation, right, the genomes are humongous. And of course, DNA methylation patterns can happen within different genomic contexts. So is it understood any correlation or relation or how it might even impact the genomic context to the persistence of those sort of genetic mechanisms?
That's a great question. So the first part, just to clarify, is that, like, you know, when a plant gets DNA methylation and certain types of DNA methylation, which I'll come back to in a minute, as long as it's in the cells that are going to create the gametes in the next generation, there is no erasure of chromatin marks from generation to generation. So, and that's why, frankly, a lot of epigenetics by McClintock and others was recognized very early, because it's you see it transgenerationally much more, where people can debate in animals how much transgenerational epigenetic inheritance happens, but it's kind of the norm in plants. And then going back to the contexts, when methylation is de novo added to a plant genome, it's added randomly to cytosines in a particular region of the genome that are targeted by small RNAs. This is a small RNA guided process. And any cytosine can be methylated. It doesn't matter what sequence context or where in the genome. But but the maintenance of that methylation then very much depends on the sequence context. Particularly CG methylation in a genic context can be very highly maintained over long periods of time, century level scales of maintenance of DNA methylation. And this is like the methylase that does this is what we call DNMT1 in plants, we call MET1, maintenance of methylation 1. And so as long as you get to that CG methylation, and if it happens to be in a promoter of a gene, it can repress that gene for many generations, if not, you know, much further out than that. And people have looked at rates of spontaneous methylation and maintenance of spontaneous methylation in plants, but it's really about getting to a certain pathway that's much better at long-term maintenance than, for example, de novo or initiation of methylation, which may only occur in a couple cells or really transiently.
So since we're thinking about rules, sorry, Keith, just a little
Mhm.
follow-up question there. What makes it so stable in plants and not in any, or is there any insights as to what mechanisms might be at place uniquely in plants?
Yeah, I mean, if we want to go evolutionarily, and this is hand waving here, the plant doesn't have the fight or flight ability to make a decision there. It kind of has to sit there and take the environment. So the idea is that you can collect information from your predecessors that are essentially constantly responding to the environment and program that right onto the epigenome and create resiliency that way. And so that's the idea. But the erasure of epigenetic information in plants. So when plants go through gametogenesis, there's kind of there's the gametes, and then there's the nurse cell that's right next to the gametes. That's important for nourishing the nurse cell with all sorts of sugar and everything else that is getting it's getting the germ cell into the right place and nourished for the next generation. And we know that the erasure happens in that nurse cell. So that nurse cell has expression of everything, this really dynamic transcriptome, probably a lot of RNA modifications, and it's generating all these small RNAs. And we think that those small RNAs are then essentially being showered down on the gametes, trying to establish chromatin modifications, trying to establish gene expression for right when it meets the other gamete, that to potentially silence transposable elements from the other gamete. We don't really understand, but there's this kind of reprogramming that's going on. But it's not from erasure in the gametes, it's erasure in that nurse cell that's right next to it.
Well, so nicely put. Thank you. So Keith, if nobody else's question, I can keep on asking, but we have a few minutes. So
I don't know if my little hand shows up.
yeah, okay, sorry. Go back to you then. Okay.
Is that okay?
Yeah, sure. Go ahead.
I wanted to follow up on that on both Connie's question and the sort of things that have been talked about in terms of asking the whole group about this question of adaptive and maladaptive priming. And so I think that what you just discussed makes a lot of sense. And I think we're all working in a variety of systems, and I'm wondering what people's thoughts are in sort of the adaptive or maladaptive nature of stress hardening and stress priming through epigenetic mechanisms and the feedbacks that we would expect to see in our systems, especially systems that may be more predictable versus cultures and agriculture versus more natural and wild. So for example, in our work with corals that have the same sort of sessile nature and can't run away, we see seasonal dynamics as you in phenotype and in epigenetic patterning. And when we look at transgenerational, or what we can look at for transgenerational, because generation times are so long, so we'll call it cross-generational, we see that those kinds of phenotypes associated with the changes tail off through time because we have seasonal changes and the same feedbacks aren't there. And so also, as we start imagining, or not even imagining, living through climate change and looking at the increasing unpredictable nature of the seasonal changes that we've known in the past and increasing storms and heat waves, what are people thinking about in terms of this adaptive versus maladaptive priming for the systems that they're looking at? So I think the coral system was very hopeful that, everything's bleaching, everything's dying, but those that don't fully die and they recover from bleaching, what doesn't kill you makes you stronger, and you kind of ratchet through these events. But these events are always changing, and they're overlaid on top of the seasonal variation. And so I can imagine in each of the systems we're looking at that we have some aspects of those things that could contribute to differences. So I would love to hear what's going on in a variety of people's systems with respect to their thinking on adaptive and maladaptive nature.
Holly, before we go deeper, I was actually going to bring up the coral point right after this because, similar topic. Would you mind speaking a little bit more about what you've done on the project so that can have a better context in what you're saying, I think, to people, if that's okay?
Sure. Yeah. Yes. Yeah. So our our project is looking at environmental, energetic, epigenetic, ecological, evolutionary linkages. We call it E5. And essentially we're using this symbiotic model. It's a nutritional symbiosis. There's really tight nutritional transfer and control between single-celled dinoflagellate algae that live endosymbiotically within the coral host cells. They provide greater than 100% of the daily metabolic energy demand for the coral to grow, to calcify, and to build the reef structure. So essentially, they're the fuel, they're the solar cells for the ecosystem engineers for the entire coral reef. And so we're using this symbiotic system because that symbiotic system is also really fragile. And we know that it can break down, and the whole metabolic underpinnings of the holobiont are then changing. And because of the linkages between metabolism and cofactors essential for epigenetic modifiers for a lot of the enzymes that Keith was talking about, our expectation of these rules is that not only energetically is it more complicated to regulate yourself, but you don't have those same metabolic modifier or cofactors for the modifiers. And so we should see environmental energetic epigenetic linkages. And that's kind of the goal of the project. And so we have multiple aims that have started out looking at seasonal dynamics, just so we understand that we already know sort of biomass and symbiont density changes through time because we have differences in temperature and differences in light. And so we're asking, how did those differences impact the epigenetic state? What does the epigenetic state look like in corals? Because they're really a non-model system with fewer genomes and less study at the genomic level and epigenomic level than many other taxa. But what we've found so far, both just prior to the study and within this study, is that through multiple species, there's evidence of, we'll call it cross-generational epigenetic inheritance, because the generation time can be years to decades, depending on the species that you're looking at. So most of the exposures that are being done for the parental generations are just able to be looked at in the early juvenile stages of the F1 generation. But this is all based on our knowledge that in the late 90s, early 2000s, corals that had bleached in one location on their tissue, work by Barbara Brown, has shown that when they received another stress event, they didn't bleach in that location, they bleached on the other side. So this memory that we've been talking about by different people seems to be found in corals. And then this was looked at across a generation by exposing brooding corals to high temperature and ocean acidification, high CO2 conditions, and asking, are there beneficial effects of the parental exposure in the offspring? And that's what we see phenotypically. We see changes in DNA methylation, and we know that DNA methylation is primarily in gene body. We know there's a positive relationship between DNA methylation and gene expression, and that there's a positive relationship between reduced variation in gene expression and DNA methylation. And so I would say what we're seeing here are these kinds of positive phenotypic effects that can carry on through time. But as we keep watching that through time, and we go through the seasons, and the temperatures get cooler and the sun gets the light intensity gets lower, and you see changes in cell densities, that there's all these external and internal feedbacks that theoretically should not propagate the same gene expression signatures, and therefore the same sort of epigenetic regulation through time, such that if you were primed to be pumping out, expression of gene A in the summer, then by the winter, if you're primed to do that, you're going to waste a lot of energy for a process that you don't particularly need. So that's kind of the setting and the context that we're working in, and trying to understand these really dynamic systems that appear to benefit from high frequency temperature fluctuations, but are very sensitive to longer term heat waves.
Great, thank you. I guess, Connie, your question?
Well, yeah, it was. So first of all, I just want to remind the webcast viewers that they can submit questions through Slido. But also then, Holly, in response to your, comment about, adaptation versus maladaptation. So I work in humans, so completely different. But, this issue of identifying rules, or just is it supposed to be like that? And this idea of adaptation or maladaptation, I think, really gets at this idea of rules and being able to predict and what exceptions to the rules would be. So I study pregnant women and, you know, trauma and stress they've endured while they're pregnant, and then look at their infants' health outcomes and look to see if DNA methylation, you know, mediates that. And we, you know, the argument is that the fetus is sort of almost infinitely plastic, and so it's much more responsive to the environment, and it can, you know, prepare for the post-uterine life, you know, by taking in those intrauterine cues. And so we think of this preparation, you know, that there's this tendency to think of this preparation or this memory as being adaptive. But that's only true really if the post-uterine environment is similar to the intrauterine environment. And so in the field, you know, we're starting to think about, well, is it really adaptive, or is that just, you know, our focus? And so it's very hard to design these experiments, but there have been a couple that have actually matched, you know, a negative intrauterine environment with both a negative and positive, you know, post-birth environment, and vice versa, to try and see what's going on. And it's very, very early days. You know, there's no data to say definitively, but at least the experiments have started. We kind of know what sort of tests we'd like to set up. And there is this openness to thinking that, well, maybe that is maladaptive, even though that hadn't been our thinking going in. So that's kind of where the human perspective is currently.
Thank you. Margaret?
Yes, thank all of you for this really interesting discussion of epigenetics. I'm on the committee, but I am not an epigeneticist. So I'm going to ask a really naive question that I've been wondering about, and I think Julie will probably be the most appropriate person to answer this one. So, how does the strategy of epigenetics dovetail with whether an animal is eurythermal or stenothermal? Is there anything known about that? Do people compare in aquatic environments animals that have broad temperature ranges, like in estuaries and so on, versus those that have very narrow temperature ranges, as to how their epigenetic strategy dovetails with that? I know it's a very off the wall question, but I've been thinking about it.
So you said my name, but I don't think I have any expertise in it. So I'm hoping somebody else does. That's not me. Yeah. Unless there's another Julie, but I don't think there is.
Yeah. No, Julia, it was because you're studying marine animals, which
I'm not.
Yeah.
Mine's an insect virus.
Oh, wait a minute.
So I think it's somebody else that.
It's the coral person. I'm sorry.
I think that's Holly.
Okay.
I got mixed up.
That's okay.
Yeah, so I think it won't be as clear cut as eurythermal and stenothermal, but I think what's more in the literature and a lot in the plasticity literature is the predictability of that change. And so if that change is very predictable, if it's wide or narrow, I'm not sure that matters as much. It probably just has to be actually sensed by the organism as something that needs to be responded to. And the more predictable that change is, the more consistent signal there is that should be responded to. And then I think the more energy efficient regulatory mechanisms come into play. So for example, in a coral example you may be familiar with in American Samoa, where there's two pools with fluctuating temperatures, and in these two pools, one has highly fluctuating temperatures, the other has less highly fluctuating temperatures, and there's front loading of gene expression in the pool with the more highly fluctuating temperatures. That essentially is a regulatory mechanism to deal with that high fluctuation. And I think the next set of hypotheses is that then there would be additional regulatory control on top of that. That's probably epigenetic, that hasn't been tested in that system per se. Because if you always need to have a certain set of genes expressed to respond to that predictable environmental signal, then you should put on some control mechanism like DNA methylation, for example. So expression can lead to methylation, and then methylation, if it's energetically efficient, could stay, or if it's not as energetically efficient, then maybe histone modifications would come in next. And there's some evidence in anemones and corals that that's kind of the order of things. And so that's another link to sort of the energetic thinking of the energetically efficient things should be happening and to hold that regulatory control in place. And in corals in particular, there's more and more literature coming out that says your environmental history really matters and high frequency temperature fluctuations really matter to reducing your bleaching response. And my pet hypothesis is a lot of this has to do with those layers of regulatory control that are keeping those sets of genes in that particular location front loaded because they're seeing a persistent signal. So I'm sort of the eurythermal, but I don't know how big that range has to be, but it's a predictability, I think.
Thank you, Ollie. Sasha, you have any points?
Thank you so much.
Oh, I wanted to, I like methylation, and I'm part of a species that has a lot of CpG methylation. But I wanted to speak up for organisms that don't have all that much methylation. So that's actually sort of, I think, second item on the slide, and I think it might be a little bit too widely or too generically phrased. Because in C. elegans, there isn't a whole lot of methylation across insects. There is a huge variation of species that do or don't have DNA methylation. So I think it actually, there is an interesting question whether or not there is a differences in responses to stress in different orders. But I wanted to make this point.
Yeah, that's exactly why I have a question mark there. So my question to Keith was gonna be at that point was what are you learning on from the high C data, because he was talking about they're running some high Cs and all. So would be really cool to also understand what other epigenetic elements then take over the DNA methylation when DNA methylation is not the key. Obviously, given the DNA methylation is the oldest known probably epigenetic modification, and that's obvious to study for a group. But yeah, understanding other epigenetic features would be great.
Yeah, I mean, I think we've created a lot of high C data. We have models of what the chromosomes and how they were interacting with themselves and with other chromosomes in the nucleus. And now the stage of the project is trying to correlate that with the phenotype and the persistence of that the epigenetic persistence of that phenotype into the next generation. I don't really have conclusions there yet, but the idea is that with DNA methylation and the transcriptome response, we've done a lot of RNA-seq that we'll see how interactions are changing and how regulation of genes or potentially suites of genes are changing and how that regulates to the environmental stress. I don't know what to call it a stress, but the changing environment, let's say. We do see much more concrete right now changes when we look at the methylome and transcriptome data. I think one that's easier for us to analyze. We have a long history of an analysis of that, but it is also pointing to specific locations in the genome that are misregulated, and the high C data is more challenging to analyze.
Great, thank you so much. Maybe next time we have this meeting, we would hear from Keith more about the other ones. Yeah, so great, thank you. I guess, so I think if I just conclude a little bit of discussion, just interest of time to keep moving to the other topics, I guess, is that one is the last point on the slide, which is the stress is likely a key determinant, and there are obviously other cues, but what we're talking from the URAL investigators is stress, whether it's a high CO2, whether it's in corals, it's bleaching, etc. And maybe in pregnant women from Connie's work, I guess, that is one of the key cues for epigenetic adaptation, and DNA methylation might be a key epigenetic element that drives it, obviously, given it's directly on the DNA, and that's much more stable than maybe in histones and other these 3D architecture, etc. So one could expect that. I'm hearing a lot from Holly also that how the transcription of genes may change the epigenetic status for it to be probably heritable more. something I guess that would be debated, but that can be debated more on. But I guess we can come back to that point if you don't have more points to discuss afterwards. But we should try to think about going to other points and having other opportunities to other I guess you're all investigators to talk about their works to get more rules of life. But I think we've derived something here, which is great already. Thanks. So the next point, the third point is the microbes impacting the epitranscriptomes in the host, I guess. And if I phrased it correctly, so I guess love to hear from the PI whose work this is. More from the virus, I think. Nobody, did I write it wrong? Where I think there was one point made into how the host viruses, hosts and their viruses, microbes, epitranscriptomes were changed with the microbes.
Yeah, that would be me. Sorry, I can't get my hand raised.
Oh, okay. Sorry, Julie. Yeah.
Sorry.
Okay.
So yeah,
So
I can tell you a little bit more about
Yeah,
this.
if you could please describe a little bit more of your findings, and then just maybe there's a point of discussion there.
Yeah. So this work really comes out of the observation that that if you have insects, and think of mosquitoes, and they're infected with a bacteria called Wolbachia, which is their endosymbiont, that those mosquitoes are less likely to be infected with RNA viruses, and they're less likely to transmit RNA viruses. And so RNA viruses these RNA viruses include really important viruses for human health, including dengue, chikungunya, and Zika virus. So there's an effort among Wolbachia endosymbiont researchers to use this to eliminate diseases like dengue. So the idea is you release mosquitoes, they have Wolbachia endosymbiont in them that prevents the mosquitoes from being infected with dengue, and if they are infected with dengue, prevents transmission of dengue to humans. And what was found was that one of the mechanisms it appears to be for why this happens, like why the presence of Wolbachia changes the transmission dynamics of RNA viruses, is actually the regulation of different RNA modification enzymes in the insect. And those are different in different insects. So mosquitoes and Drosophila are kind of the two systems that it's looked at in, and they do respond differently, and it's different depending upon the Wolbachia as well. And so what my collaborators showed, which includes a virologist and a person who works on Drosophila Wolbachia systems, is that it's actually, at least in part, due to the RNA modification. So Wolbachia, I'm going to talk about Drosophila, but Wolbachia and Drosophila modifies the expression of DNMT2, which in Drosophila is thought to modify RNA with 5-methyl-C. And that in turn actually modifies the RNA methylation profile of the virus. And that in turn changes the infectivity of the virus and the persistence of the virus in the insect. So it's a complicated system because there's 3 members, and it takes all 3 members for the biology to happen. But that also makes it fun. And I think potentially this is, like, with respect to the rules of life, this is possibly a mechanism by which microbes can alter their hosts, but also alter other microbes that are in the host as well.
Sounds good. So I have a question before others do, I guess. How do they do that? Are they secreting specific metabolites that might be inhibitors to histone or epigenetic modifiers, etc.?
Well, so they're definitely changing, like the presence of Wolbachia changes the transcript abundance of at least where we know about it with DNMT2, it changes the abundance of DNMT2. And then DNMT2 is thought, in Drosophila, you can do all sorts of genetics. So DNMT2 is then thought to be the writer that then changes the methylation profile with respect to cytosine of the RNA in the virus, because these are RNA viruses.
So I think that was the tRNA modification, etc. That was from Tim Bester's group and all, right, from DNMT2 base. But okay, yeah.
And then there's also other modifications as well. So we do know that there's I think we know there's pseudouridine modifications, and we know there's 6-methyl-A modifications as well in the viral RNA.
Got it. Okay. Other questions for Julie? This is just understanding how the different microbes or things that infect us might be modifying the host epigenome. Okay, well, no, then we'll move on to next ones. I guess, thank you, Julie. But I think one important rule of life, just to summarize, would be how we should understand how external organisms might be communicating to the host epigenome by directly modulating it or through something else. And that might be a more novel area of epigenetic communication between different, I guess, biological systems, whether they're foreign or with the host. Yeah. And something that's is really cool, sounds really cool to me. Okay, thank you. But the important piece in disease biology as well. So we should move on in the interest of time to other participants as well. And I guess the other thing I wrote about here was how different RNA modifications potentially can be read by the same reader pocket. So this was probably on the YTH domain. If the PIs here, love to hear from them.
Yeah, so I'll definitely get to that now, but I was really wanted to, if I'm allowed to ask Julie a question real quick.
Sure.
So Julie, we think a lot also about environmental factors, of course, when it comes to the RNA conversations, RNA providing more rapid transcriptional responses, temporary sort of adaptation mechanisms. So one of the things that I'm wondering when I hear about your project is how much has it been differentiated or looked into in the field that these contributions are direct versus indirect by the organisms providing any changes to the environment that then influence sort of the host. Just kind of wondering like where thoughts are in terms of coming up with the quote unquote rules.
Yeah, I mean, so of course an endosymbiont will change the environment, right? Like it's just that by nature, right? Like, so if you take an insect with or without an endosymbiont, you essentially are changing the microenvironment inside the organism. So you can't rule out that it's not that. But I mean, if you have the same insects in the lab under the same environmental conditions with and without Wolbachia, we can see these differences in the virus and the methylation patterns on the virus.
Yeah, so I guess I was thinking even broader, like when you bring this to out there, like an ecological model, where then you have thousands of insects and you could even be producing, changes to the ground or to the climate, like big scale, just in terms of bridging scale. I'm just wondering if there's, I don't know.
I think bringing it globally is actually a challenge in many respects, because yes, insects, there's tremendous diversity. There's tremendous diversity in the Wolbachia endosymbiont. And then you'll even have viral diversity as well. And of course, they're all going to be adapting. Like when you release one Wolbachia endosymbiont, it's not exactly in an insect, it's not exactly the same a couple of generations later, much less, a couple of years later. So I think, there are issues with that. I think from the endosymbiont side of the world, like we're kind of used to that in terms of there's a lot of diversity. I think that connection between how much of that diversity is really due to the epitranscriptomics, like we don't have any feel for at all, right? Like it's too new. Like this is just too new. It's too new of a finding. I think one of the reasons I was brought in to do more of the like ONT development of the ONT epitranscriptomic sequencing and analysis is like, typically it was easy to look at epitranscriptomics, or well, not easy, but typically epitranscriptomics has been done on ribosomal RNA, or looking at RNA modifications has been done on ribosomal RNA, tRNA, and viruses because you can purify loss of material. And I think one of the questions we're asking is, okay, what does that look like at the mRNA level? Because we can now do that. Like we couldn't really do that before with techniques like mass spec or other techniques because it wasn't enough of those RNAs. So being able to look at it more globally. So I don't know. I'm not sure if that answers your question, but I think we're trying to kind of bridge and understand it, but it's still really a new area in many regards.
Great. Lydia, could we hear more from you about your work? And I think we have then Jeff and Kaushik to cover as well. Want to make sure that everybody gets time and, we all are discussing
I saw
science.
Holly had her hand up. Do you want me to?
Oh.
I
Holly,
don't want to talk too much, but I wanted to follow up on that as well, because I think that's this question of like, how do we scale up when we have symbiotic systems? Like we're really in that area as well, because we have this holobiont of the host with its dinoflagellates and its bacteria and its viruses and its fungi and all the things in there, and creating a different physicochemical environment depending on morphology. And so I want to, and also going back to our question or discussion on like the environmental signals that need to be perceived and what is stress, like are other people looking and asking the same question of how are signals perceived within the symbiotic environment? What is it that matters, the external environment or the internal physicochemical environment? And the same kind of question of, is this a direct epigenetic modifier that's passed as information, or is this a physicochemical stimulus from a metabolite changing the pH of the internal condition? And then this other question that the topic that Julie brought up of generation times, right? So your symbiotic system is, depending on what your components are, some are going to be evolving and adapting to be a different symbiont, but the same sort of like tissue that they're living in of the host. And so I don't know if Lydia, you're going to talk about this too, because you're asking the question, but I'm also really interested in that, like how are these signals actually being perceived in a symbiotic system? And I think without being able to understand that, it's hard to know what the rules of sort of signal transduction to regulation are.
So I guess I'll go next. Those are great points, Holly. And we think about that a lot in the overview of our work, because to tell you a little bit about our project, our main interest is that intersection between environmental chemistry, sort of physiochemical properties in the cell that change, so that interplay, and ultimately how that translates to molecular properties like sensing, right? So what is a signal? What is the threshold? When does it go away by these interactions? So the observation that really leads to this particular project is that they are now that it's pretty understood that the notion of chemical modifications on nucleic acids happens in many organisms, and there are many processes that go along with it. The recognition, so the reading it, the potentially deleting it or erasing it, as well as the actual putting it in there, the writing piece of it. So we've been kind of thinking about that. And then with that came along a lot of the observation that there's a lot of proteins that are being uncovered, and that a lot of these processes are covered by proteins in cells, in different systems. And along with that has come crystal structures for some of these proteins, and observations that they are binding pockets and motifs that do have things in common across species. So we kind of really have been collaborator with a great computing kind of group that thinks about chemical properties in molecules. So Dr. Tamamis and his lab at A&M. And my students and I really took this opportunity to ask the question, there's so many modifications, there's so many proteins that are being uncovered, some for which we have sequences but not structures. How can we take this into a creative way and come up with a high throughput way by screening, coming up with patterns, coming up with biology that we can then test in the lab? So that's the premise of the work. It has started with pretty much understanding structures that are there. So YTH is one of the models we're using because there's at least 5 of them that are different, that have different lengths, different sequences that we can use, model their structures. They have enough homology that we can start studying, we can start searching for things like it in different organisms and really start to screen for the first question. Do they recognize the same things? How flexible are the pockets? When you're thinking that more and more hundreds of modifications are being understood, granted, we still don't know that all of those are functionally relevant, but the question really becomes, cells are extremely efficient. Do we really believe that each of them has a dedicated protein that would recognize them and process them in a cell, et cetera? So this idea that there could be some promiscuity, some flexibility at the molecular level that can enable multiple pathways and chemistries is something that we've been really interested in. So we started with very known methylations, so M6A, very good, well-known motifs that recognize it, where it's understood that there's a lot of sort of aromatic chemistry in those pockets, and really wondering how flexible, how common, how widely distributed across organisms is this? And we're starting to really understand that there are some basic rules in the chemistry, but there's also quite a lot of flexibility in those rules. So those deviations have become very interesting into what they mean as far as biology. So that's the project that we have going on. It's really a good opportunity with this excellent group in terms of really high throughput data to give us patterns that we can then test.
Great, sounds good. So you're finding more concepts on probably how reading these epitranscriptomic modifications may occur, right? Is that correct?
Yeah, that's what we're doing. So we're trying to find really patterns that are common to that specific chemistry that are also leading to a lot of understanding of specific versus unspecific the RNA protein interactions. And again, the main motivator is to really start thinking of these modifications in the context of agents in the cell that interact with them. And we know that proteins can modulate a lot of the dynamics.
And is it specific to methylation, you think, or there are other kinds of modifications that may occur in RNA, may have probably different rules, because we're talking about rules of life here, yeah?
Yeah, so that's a great question. So we started with screening all modifications that are known for which we can actually have energetic profiles in computing and in silico. So we're not limiting our screening. We're really asking the broad question of, we suspect you look like an M6A recognizing protein, but who really are you? What else do you really recognize? Is it the same binding site? Is it the same pocket? Can your pocket fluctuate and make room for different interactions? So we're really doing broad in silico searches to start.
Great, thank you. And how does it compare to like histone modification reading, for example, which obviously is a big field and quite solved, I would say?
So I don't know that we're using any proteins that are specifically devoted to histone recognition. It's a good point. We can look more into that.
Cross comparing, that would be good. I mean, from again, rules of life perspective, right? I mean, larger picture perspective. Yeah, sounds good.
And even from the perspective of, are these, what rules make these more biased towards RNA than DNA?
Absolutely. Right? Absolutely. Yeah, agreed. And is there specifically different kinds of RNA eventually? Yeah. Other people, questions for Lydia? Oh, okay. If not, then sorry guys, I will have to leave because I have to go to an ACS grant review right after this, like starts in 5 minutes. So Sasha will take over this part from here on. So, but thank you. This was a pleasure to be moderating this session, at least until now. Sasha, over to you.
Thank you, Kunal. So we don't have to stick to our schedule too precisely. So I think that scientists in general like to talk about science more than about challenges in herding cats. So in that spirit, I think Jeff and Kaushik didn't have a chance to tell more about their work in this program. So I think Jeff has microphone ready.
Yeah, well, I can actually talk about herding cats simultaneously with the actual science, so maybe that'll be advantageous. So it's kind of interesting hearing everybody talk because our project is quite a bit, I think, maybe less mature in sort of the state of the biological system compared to a lot of the projects that have been described. We're working on bumblebees. And in particular cold tolerance. And bumblebees are kind of interesting because unlike a lot of other insects, they're capable of facultative thermogenesis. So they're sort of halfway between an endotherm and an ectotherm. So they can generate heat actively through shivering. And they have, thick fur coats for staying warm. And so they can be active in quite cold temperatures, but there's also populations of the same species that don't live in cold environments. And so we have several species that range across these heterogeneous landscapes. And so we are actually getting back to that point of sort of variability that was brought up early with different sort of thermal mechanisms. We have populations that live on mountaintops of certain of the species that we're working with that experience extreme shifts. So summer temperatures can get as high as, anywhere else, but the winter temperatures are extremely cold that, early season individuals experience as well as overwintering queens compared to those individuals at lower environments. And so what we see from the physiological perspective is that, at least our evidence suggests this, is that individuals that are derived from queens that are reared up in the lab from high elevation environments have much lower thermal tolerance limits, so they're able to withstand colder temperatures than individuals from reared up from warmer climates. And so that sort of was the foundation for this work, and we showed up fairly convincingly that there was a fairly strong correlation with sort of the natal temperature of where we collected queens and where we rear these things up in the lab. And so I don't want to get too much into the project because we do a lot of the same stuff that's been discussed already with the DNA methylation, but in response to stress, as well as transcriptomics in response to stress and trying to figure out cross correlations between these different levels. But there are a few interesting things that are sort of unique about the system in that we are sort of trying to get at that variability. So those populations from high elevations that have greater variability in environmental conditions also have a broader thermal tolerance, right? So the critical thermal maximum doesn't change very much across environments, but that minimum varies, right? Suggesting that there's some aspect of variation in the cold tolerance, whether that's plastic, adaptive, epigenetic. That's sort of the idea behind this project. The other thing that's interesting is the idea of maladaptation versus adaptive, because some of the things that we see is that maybe in a lot of insects this is true, is that upper thermal limits don't seem to vary very much, right, across large geographic gradients, but lower thermal limits do. And so one of the problems is that upper thermal limits are going to get tighter and tighter and tighter and tighter as climate changes. And so if organisms are adapted to the cold environment extremes, which may be the case for bumblebees, that as that temperature increases and there's a cap on the upper limit, that cold adaptation may actually end up being maladaptive. And so there could be some conflicts as genotypes start shuffling around. And that's sort of some of the things that we're working on. In terms of progress, I will say, because again, some of the questions are very similar to things that have already been discussed, and I don't want to be redundant. But the challenges have been extreme, right? In part due to climate change. COVID eliminated a field season, so our work is very field based, right? We have to go out every year and get fresh queens to rear colonies in the lab, which requires cross country field work. This year we camped on Mount Hood for a month, waiting for the queens to come out because the temperatures were quite cold. So that was fairly dramatic. Last year was extremely dry, and so the number of individuals that were out and about were very low. So we've had a number of challenges that have been driven by the field-based requirement aspect of our project, in addition to loss of a field season from COVID. So that's been a really interesting challenge to try to overcome. But this year we finally got everything I think that we need, at least to do most of our project. But we've been a little delayed on the actual epigenetic inferences, although there have been a few interesting things. We do see sort of what's been shown before is that the sort of very parallel to the corals, right? The methylation is highly restricted gene bodies, and in fact, it's largely restricted to exons, a little bit in 1, but mostly 2 and 3, and then it declines again, and then it spikes again at the tail end of the gene. So we're trying to figure out, why that might be, and it's pretty consistent across individuals, but we do see variation. Individuals before and after stress do have changes in their methylation profiles, as well as changes in their transcriptional profiles. And so these are kinds of things that we're still working through at the moment. And part of the challenge has been, because of delays in the field work, a lot of our research lab timing got decoupled, right? So the chemists who are doing metabolomics work and the computational biologists are sitting around like waiting for the data, to come through, because so that's been a major challenge, is just the getting all the time points sort of worked out so that everybody has something to work on. But I think we're finally at that point now. But that's been my biggest challenge as sort of the lead PI trying to herd cats, is trying to get everything temporally organized, because everybody's doing different stuff. Keeping the bee people organized, I said this in my response, has not been that big of a challenge because we've been working together for a decade now, those PIs, but we have several add-on PIs that are the more interdisciplinary components, the chemists and the computational sciences, that are interested in the project but aren't quite as invested as the bee people have been. And so that's been one of our biggest challenges, for me, is just to make sure that everybody's aware of what's going on, because it's in my brain constantly, but it maybe isn't at the forefront of all of the collaborators' brains constantly. And so that's been my biggest challenge, but it has largely been driven by the difficulties of working with a field-based non-model system. So I think that's all I have to say, because everything else is sort of redundant.
I have maybe a couple of quick questions. One, I was just curious. I assumed that was from you because it was about bumblebees.
Probably
in anonymized list, but it mentioned that you were looking at variation between populations. So I'm curious. First of all, how much difference in the regions did you find across populations, and was it more due to loss of cytosines, or was it, you think, regulatory variation that changed it?
So this is kind of interesting, because we've analyzed the data both ways, and both are interesting, I think, right? Because so you could have loss of cytosines or guanines, I guess, but you can lose the actual base, which can lead to a change in epigenetic profiles, or you could actually have the demethylation itself, and both of those could potentially be interesting when you're thinking about a large biogeographic scale, right? Because populations accumulate mutations and so on and so forth, and so we've analyzed the data both ways, because we're working in a system where we actually have a fairly comprehensive 700 individuals have had their whole genome sequenced. So we have a pretty good idea of where all the common SNPs are in the genome, and we can actually remove those from the data set and compare both ways, basically. And so it's both. We do see that there is methylation differences between populations across the range, and it does seem to be somewhat clinal, suggesting that there may be sort of a population structure to epigenetics. that's similar to nucleotide variation, but it's not entirely, or at least it doesn't seem to be entirely driven by nucleotide variation, because when we remove those sites, you still see variation present.
How do you distinguish between the population's sort of a genetic structure and the fact that they probably have different environmental conditions?
Yeah, so that's sort of the key thing. And so for the data that we've actually analyzed so far, it's not really been possible, right? So that's sort of, we're just seeing, is there variation, right? And so that's the first step. But for the actual main project samples that are being analyzed currently, we actually have multiple mountains that we've sampled, low and high elevations, from different altitudinal transects. And so the idea there is to try to somewhat and this is all in the main project samples are all within the state of Oregon, so that they're within a day's drive of one another for the most part. And so the idea there is to try to decouple the space and environment aspect by replicating the mountains. The other thing that's interesting is that bumblebees don't have a ton of population structure. When you look at the nucleotide sequence, you definitely see like an isolation by distance effect, but it's extremely low within a species in most cases, unless there's unless they're really, really alpine adapted, and sort of you end up on these sky island populations. And so the ones we're working with are not like that. So they're very homogeneous in terms of their genetic structure. And so that's sort of one of the advantages, right? Because you can sort of get at, if there are epigenetic and transcriptional changes, there's sort of something's going on independent of a lot of the population structure and nucleotide variation. So that was sort of the idea, but it's definitely there, but it's lower than a lot of other organisms I work with.
Thank you. Yeah, I think that the sort of eusocial animals are really cool from the epigenetic point of view.
Yeah, well, that's what's interesting about this, is that a lot of the epigenetics work that's been done in the eusocial animals has been with like task determination, task orientation. So like, what tasks are you doing as a worker versus a queen, and whether there's any differences between, say, bumblebees and honeybees. And I think there's still a lot that's not known. There's a lot of debate still about the role of epigenetics, even though that was sort of the DNA methylation was initially thought to be a really, really important. I think there's still some debate about that. That's not really. I try to stay out of the social stuff because it's fairly packed with researchers. So I'll leave that to the social experts.
Okay. So if there are no immediate questions for Jeff, Kaushik, would you mind?
Sure. I'm happy to take on the burden of redundancy going as the last person in this group. So hopefully I don't keep saying the same thing over and over again. My lab also works in one of the least social of organisms, which is a single-celled microbe, which is Schizosaccharomyces pombe, which is yeast. So it's been really interesting to hear about the more complex systems that many of you have been talking about. The only comment I wanted to make was about this question of maladaptive versus adaptive, because everyone's been thinking a lot about this. I wonder if timescale should really be a question that needs to be considered in considering maladaptive versus adaptive, because some of it is anthropomorphic, right? I mean, in the sense that organisms make a change. We know it's maladaptive because we can't look into the future, but then when time passes, whatever the time scale is, maybe we have a better understanding of what adaptation actually looks like. And so we work in organisms that divide on the time scale of a few hours. And I think the project that I'll describe very quickly has a lot of overlaps with what Keith talked about in his system in plants. So we're very interested in understanding how the misregulation of an epigenetic modification, which is called heterochromatin, or it's an epigenetic silencing mechanism in yeast when misregulated misregulated leads to the acquisition of novel adaptive traits, meaning then when you misregulate the system, cells don't really like it, but somehow there's a novel epigenetic island that shows up that enables the organism to adapt. And once adapted, it can convey this adaptive state heritably for many, many generations. And so we were really puzzled by this. And so we invented this approach of actually being able to design this trigger for epigenetic adaptation in a very precise manner using oxygen-inducible degron approaches so that we could actually follow how adaptation happens. Essentially, we wanted to do what I was talking about earlier with this adaptive versus maladaptive is like, can we watch the scope of adaptation that happens as a function of time? In this particular case, we were working with a very tractable genetic system, so we could trigger epigenetic adaptation. And then we could use RNA-seq and ChIP-seq and many other kinds of whole genome analyses approaches to actually follow all of the spectrum of choices that cells actually make before they actually conclude at this one adaptive choice, which seems to make them fit enough to actually continue to grow and survive. So this is one sort of area in the lab. I mean, this was really the major focus that sort of brought together this team of researchers in this URAL project. And this is one of the conclusions that I wrote down, which is that, what we found is that stress actually is a key determinant of successful epigenetic adaptation, meaning that we found that stress pathways actually have to get activated for cells to actually conclude what genes need to be activated or silenced during this adaptive process. And of course, there's this philosophical consideration of stress, right? I mean, that emerges out of it is like, oh, I mean, is being stressed always a bad thing? But here was an example of an organism that could actually successfully adapt only when it underwent the activation of the stress response pathway. As part of actually having this time-dependent approach where we could follow epigenetic adaptation, essentially watching every decision possibly that this organism made as a function of time, we also used single molecule imaging, which is just a biophysical approach of actually watching proteins move inside cells. And the motivation for this was really to see if we could watch complexes actually assemble within the cell. So essentially, could we take biochemistry that we can do in test tubes and watch proteins interact with each other, but could we actually watch this happen inside of a cell? And would that tell us very different rules from what we actually learn about biochemical interactions in vitro? And this was really motivated by the fact that if adaptation is a process that involves the relocalization of epigenetic complexes from where they are supposed to be present all the time to new places in the genomes in order to enable adaptation, then we needed approaches that were able to actually watch the dynamics of many of these proteins within cells itself. And in this process, we identified the second rule that I try to highlight, which is really, we think of proteins as actually forming complexes A and B, and then traveling to chromatin and localizing and performing some function. But what we really found that's happening inside cells is that these complexes form only on chromatin, meaning that chromatin, which you think of as an inert scaffold where things actually float around and ultimately land and perform their functions, is not actually inert, but is in fact a catalyst that actually enables complex formation between multiple factors. So when you think of many of these epigenetic processes that many of you are talking about, these are absolutely large multi-protein complexes that are involved in gene activation or repression. And what we're finding really through single molecule imaging in cells is something that the biochemistry in vitro was not telling us, which is really that chromatin, which is inert, is not actually inert. And the fact that it is an active catalyst that enables complex formation. So these were really some of the rules that we identified. And I think what's really cool about the system that I think has propelled the URAL project forward is ability to watch adaptation as a function of time. So essentially, we're able to both fast forward and rewind the adaptive clock in order to figure out how epigenetic adaptation works.
So in the interest of time, I will probably cut the question and answer sort of round a little bit short. And thank you, Kaushik. So what would be before we switch to looking at the sort of challenges and achievements in trying to deal with the multidisciplinary teams, what do people think about the sort of what the common themes that emerge? So one sort of very generic 30,000 foot view is that there is sort of across different projects that people talked about across different organisms and different sort of scales was that sort of a general focus of adaptive or maladaptive role of epigenetic response, which could be DNA methylation, or it could be three-dimensional structure of the nucleus, sort of in response to various stresses and primarily environmental stresses. So that's sort of a very generic formulation. So what would we want to add to that? So I'll leave this question open and maybe ask for the next slide. As you will see, there is a very common list of themes that actually have been sort of recurring through the responses of the PIs. And some were sort of out of anyone's control, like COVID disruptions, and we all had to learn to Zoom and everything. So the recommendation of that is not have pandemics. But more sort of generally, sort of the positive lessons learned were that there is a more and better interactions are key for successful work interdisciplinary, especially, and key words that were mentioned are mutual trust and proximity, and sort of having regular interaction. Of course, that's very important. And one sort of metaphor or idea that I thought was really great was to a translator scientist. So one question that I wanted to pose, sort of looking forward to the participants, is: do you think, as far as training people and sort of managing multidisciplinary teams, is it the idea to would it be better to have everyone become more of a polyglot? Or is the idea to try to have more sort of translators who can sit on multiple chairs and translate between, sort of roughly speaking, mathematicians and physicists and bench and field biologists? Another sort of more general, so the challenges are partially organizational, and they're kind of the opposite of what was going on. Actually, Holly had a, did you have a point?
Yeah, I was just gonna speak to the sort of translator scientist, and that's worked really well in our group. That was our response to this, is that we partially strategically designed it that way to begin with in the proposal, because of the often lack of common vocabulary across the groups. That's made it really difficult. Like, you just spend a large proportion of the grant just figuring out how to communicate. And so we particularly picked people who had worked in modeling but had trained in symbiosis, or these kinds of positions, so that we could really think about how we get across these different scales. And I think that that's something, with another point on the slide, that I also wanted to say is, like, the translation between the different teams, because for me today has been super exciting and stimulating to see what the other teams have been able to do, and to be able to even translate between the different teams, and have a centralized place to share ideas and share emerging rules of life and rules of life. And I think importantly from our system is really, we have our model system, but it's really, really non-model. compared to what a lot of people are working with and compared with what actual model system field is possible to do. And so I think the technology and methods transfers between the funded groups would be super helpful in having translators across what's possible in science, even if your particular system isn't quite there yet.
Thank you. Connie.
Yeah, so it really strikes me that one of the key things is just you need more time. If you're pulling together different groups and you're having to learn new languages and concepts. And I mean, I'm a total advocate of multidisciplinary research and that you learn more things that way, but it absolutely takes more time. And, I think that just somehow has to be baked into, the proposal. I'm sure we've all requested, no cost extensions. I'm kind of curious how, what the maximum number of no cost extensions is that have been approved, but it'd be even better if it were baked into, the time frame of the grant. And another thing, so that people talk about, getting the translator scientists and get training in other labs and, even workshops between labs. And I just applied for a big center grant from the American Heart Association. And so they have that every proposal has to include travel funds to, not only have PIs and postdocs traveling to other labs within the proposal, but that the different centers have to have travel funds to travel to each other. And it's even baked into the review process that the second stage of review has, sort of the finalists at that point all come together for a meeting, and people that they do some sort of evaluation on how collaborative, how interactive, how sort of stimulating, the PIs from the different grants are going to be, which, I'd never encountered anything like that. I hope we make it to the second stage so I can see how that actually works. But that seemed to me to be very supportive of what you need to pull off this kind of multidisciplinary research.
And so these are really tough challenges. Lydia?
I just want to quickly echo what Connie said. I think many of these projects started in the middle of the pandemic. Literally, we got a call when we were not even with our students. Students had a lot of things they had to manage, just like faculty. And I think that when you're working with multiple teams, multiple languages, multiple disciplines, that standard NSF 3-year project model might be something we want to rethink about.
Okay, thank you.
Can I add to
yeah, please.
to the end of that? Sorry, I didn't raise my hand, but I think especially, in light of the pandemic, I think where a lot of laboratories went, and this was particularly tough on interdisciplinary projects, where a lot of laboratories went is they went back to their bread and butter. We got to make sure to do the things that keep the lab alive, these core research projects that all these different collaborating labs have. And I think that a lot of times, and I think Jeffrey mentioned this in his comments, I think a lot of times those laboratories, when they focused back on the core, for a while, that other thing they were doing, that interdisciplinary UROL project became this other thing that, was just kind of just starting out and kind of got thrown by the wayside. So for me, there's a point on the slide that's up right here that's considered giving lump sum funding at the beginning. I would highly argue that, and I think that it was difficult to see a lot of people in the beginning using a lot of the funds, even though, for example, they were on data analysis teams, even though no data had been produced yet. But they needed that during the pandemic. They needed that early on to finish up maybe other projects. And I think there's to some extent been, I've had a hard time getting the group focused on this project, as it's everybody's kind of side thing, besides the lead PI, which is, has my name down and my reputation at stake. So I'm the one really pushing for it, but to an extent there is a herding the cat aspect.
Right. So we are behind schedule. So Kaushik, I wanted to Yeah, go ahead.
Yeah, I just wanted to echo what Keith was saying and others also, and just think a little bit. I mean, from the perspective of the URAL grant, especially when it's an interdisciplinary team, I think we're not farming out things to collaborators, right? I mean, this is not a company side kind of model. I mean, as academics, I think the training aspect becomes very important, and so people have to fundamentally, I think, be trained in different disciplines, and that would be the meta point of actually having interdisciplinary teams. And I think that the time aspect of it does become extremely important in this. It is actually a really new way of trying to think about doing science by actually trying to build multidisciplinary teams. But I think the resources, I think time is an important resource in essentially making or realizing the full benefits of such a team itself.
Thank you. So I think it's fair to say that sort of wanting more time is important. So Lydia, you have a question?
Oh, sorry, I just forgot to lower my
Okay.
hand.
Okay. So Connie, do you want to quickly touch base on the societal? Okay.
Yeah. I'll just take 1 minute and then we'll take the
Okay.
break. Could I have the next slide, please? Okay, so just pulling out from all the different responses, something the things that were clearly societal impacts were, develop projects or products to help communicate climate change to the general public. There was something specifically through targeted to K through 12 education. The question, will increasing carbon dioxide and a changing climate have a long-term impact on crops and agriculture? Then third, our ability to forecast impacts on coral reefs, and also what have we learned about how to better conserve and even restore coral reefs? And then fourth, how can we manage host microbe interactions to benefit human health and agriculture? So these are the really big picture ideas that we can communicate to the general public and, taxpayers, that their tax dollars are being well spent and all of that. And I mean, I think these all sound great. And I guess the idea is that we do want to continue to think about this, because this is how NSF communicates that the money's being well spent. And I think these are all great examples. And I'm sure, we all could think of things like this. So any final comments on before we close out this, the interactive discussion component? Okay, so then we've got a 10-minute break scheduled, and then we're going to come back and we want to quickly develop some consensus on common themes. So, what are some of the rules of life that are starting to emerge that we think can be used for hypothesis testing in the future, for future calls for, grants? So if you guys could maybe think some about that. And so I've got like 27 after 127. So let's try and come back like, 135 or something after that, so we can still end at 2 o'clock. And so then the people who will stay will be the moderators and the NASEM staff, and discussants, you can go on bathroom breaks or a cup of tea or whatever. So see you back in slightly less than 10 minutes. think so. We've got Keith, Kaushik, Holly, Jeff back. Let's see, is Lydia back? Julie? So I think everybody but Lydia's back. Do you want to go ahead and start, Sasha?
Right. So basically, and I think that she's back as well. So basically, this is the partially called from the previous slide, partially notes on what we were discussing in the previous hour, and there is a link shared to everyone in the chat. So the idea is to sort of help formulate things so that they and help Connie in sort of synthesizing what we try to do. So please speak up or just directly type in the shared link. But one sort of there are specific advances that are that have been shared on slides and also on what Camila had been capturing and writing down on the next slide. But I think that one, let me try to reformulate it, is epigenetic responses play adaptive or maladaptive role. And it's across scales and across species, but it looks like a very common theme is how organisms and ecological systems respond to stress. Another point is that this is probably a useful point across different workshops, and that was one of the of different programs in your role overall. And of course, our views on what adaptation or maladaptation is are likely to change as we get sort of deeper understanding of the systems.
Sasha, just so our view of adaptation or maladaptation may change over time, which is what Kaushik brought up, but then could also change versus how we use it in terms of what the application is, like what Keith was talking in terms of, breeders using this increased growth with increase in carbon dioxide.
Mm-hmm. Okay. That is so do we want to add, so it's a little bit difficult to sort of a bridge sort of a specific findings that we were talking about with sort of grand sounding rules of life. So do we want, or what do people want to add to this, or do we think that this sort of captures the general sort of tenor?
I think we should try to add some more. I mean, we want to.
Yeah,
I think that there's a lot of research. I think we want to show, the success of the URL program and all. During the break, Carol was talking about Lydia, what you were talking about, flexibility in the system, and I can't remember your exact wording, but maybe Lydia, you could help us, word another point here under rules of life.
Yeah, Connie, it's sort of like, what I was saying is, so the rules and then the flexibility built in within the rules that allow for diversity mechanisms,
Yeah.
right?
So to get a little more specific, so could you say something about, based on your research, not just that what a rule might be, but that some flexibility to the rule would be baked into the system.
Yes.
To allow for more flexible responses for
Yeah,
future
and
challenges.
that flexibility could be as a result of, a stressor. It could be as a result of different interactions, as a result of something dynamic about the system that sort of allows the system to be flexible, expand, or break the rule. Rule breaking.
Holly.
Yeah, I think this idea is really important. When we've all talked about, not all, but there's been a lot of talk about DNA methylation, and there's talk about other more complicated or less well-known mechanisms. And I think there's been in the literature this idea of inherited gene expression regulation, where there's a rule more so about the fact that there can be a cellular memory, as opposed to it has to be DNA methylation, or it has to be this exact sequence of events that's leading to this outcome. So I think it's really thinking about the rules that could underlie multiple mechanisms, or multiple mechanisms that could end up in the same outcome, even if their exact stressor, magnitude, timing, et cetera, is not the same. So I think there's been a recent review paper by Adrian Kulchhauser et al. That have recapitulated this idea of inherited gene expression regulation brought about by multiple different kinds of epigenetic mechanisms with different timings that give us some of this flexibility of the rules, I think.
I think it's a really important point to make, that these are sort of memory mechanisms with very different time constants. So that's Julie, you hand raised.
Yeah, so what I was going to say was, I feel like some of our experiences with endosymbiotic viruses and insects and the diversity of them and the diversity of their responses kind of goes to that same dynamic responses and flexibility in the rules, but kind of at a different level in the scale, right? So instead of at the protein level, really at the organismal level, you see the same thing.
So does this capture also like organizational scales?
Yeah, maybe.
As in or like protein, or like molecule to organism.
I mean, and probably even to communities and right, like, because I guess we even have communities, really.
Mm-hmm.
So
Connie, yeah, go ahead.
I think it might be interesting to say something about, DNA methylation or epigenetic variation in more generally across species. What Keith was talking about in the beginning, why DNA methylation seems to be more prevalent, more of a player in plants, possibly because they can't move. That's a really important point. And, so since we've got everything from, Pombi on up represented here, it might be nice to try and formulate something about the role or the rule of how epigenetic variation is used across different, different organisms, different levels of organisms, and why that might be. Who could start formulating something on that?
I like this. So that's my attempt to start writing something about this, that the specific mechanisms can greatly vary. There is a lot of epigenetic variation. Kaushik, please go ahead. And actually, because I have one eye on this, well, don't wait for me to say go ahead. Just if you want to say something, please go ahead.
Yeah, I just wanted to respond to Connie's prompt and consider the fact that with unicellular organisms, I think there's a lot more exploration that's possible than there is with multicellular organisms for solutions that actually might be adaptive in the sense that every cell in its is a universe of its own with unicellular organisms, as a result of which if you consider this as a sampling problem, each cell can actually roll the dice in a way that multicellular organisms cannot. So I wonder if the flexibility of solutions that we've been talking about is just much more prevalent in unicellular organisms in ways that requires coordination across tissues and cells in other higher eukaryotes.
Okay, so that's really interesting. So let me make sure I can wrap my head around this. So you're saying unicellular organisms might have more flexibility in experimenting with, different roles or different variation in epigenetics and DNA methylation and what it might do?
I mean, not to center it on the organism itself, but just that as a population, many of these cells can keep making very different decisions, but the population as a whole will survive as long as a few of them actually arrive at the right solution. So in terms of epigenetic adaptation, the chromatin, which is the substrate for much of the epigenetic processes we've talked about, is all across the genome. So everything looks roughly the same. So when cells have to make decisions about what genes to activate or silence, how do they actually do this? It fundamentally comes down to some sort of a sampling problem. And sampling with a unicellular organism just means if 1 million of them die, there's still 1 million of them left behind.
So Julie and Holly, don't wait, just jump in.
So I was just going to raise that, like many of these, and maybe the discussions also have focused really on epigenetics and kind of the adaptation. But I mean, I would say that like our research doesn't really fall into that, right? Like, because it's epitranscriptomics, arguably maybe adaptation. I don't know. I think it's a reach. But so I guess I was just going to say that I would just, I would encourage us to expand like the breadth of our advances and rules of life, because I think we have a lot of stuff represented here that isn't necessarily epigenetics and DNA. But I don't know quite how to do that. So I'm putting it out there, but I don't know what should be, like what would be a way to fill that other than something very broad that wouldn't be helpful.
I wanted to follow up on the variation across organisms, and can we get rules across taxa or across phylogeny, sorts of questions. And I think it's the sampling of the unicellular organisms. I think it's a really interesting idea. And I think also on the sort of same side and flip side, their sampling of the environment and what the external environment versus the internal environment is like with, across different taxa with different life history traits. And that's kind of what we talked about a bit today with, symbiotic, non-symbiotic, what you're sensing, how much of the environment you're averaging over versus what scale or fine scale do you sense of the external versus the internal. And that could lead us to a place where we think something is adaptive or maladaptive for what we're measuring and perceiving as humans from the external environment of in my case, temperature or CO2 or ocean acidification or salinity change or something like that. But the cell is buffering that and maybe only getting some fraction of that. So I think it's, when we think across these different taxa and we think about single cells, and up to very complex organisms and the different ways that they transduce these signals will then trigger their regulatory responses, I think, in different ways. And so there might be clustering or clusters of taxa that are doing similar things because of the way they're maybe transducing, sampling the environment or being sampled by the environment or selected by the environment.
So I think I'm trying to capture this idea by saying that the idea of what is adaptive or maladaptive should involve considering specific environment at a given scale. So like a given cell in an organism behave, or like has different environment than the whole.
What that cell or what that organism is actually experiencing intercellularly.
Right. And maybe I love the idea of sort of looking at epigenetic mechanisms as part of a general, like more general phenomena. And one way to think about this is to think about sort of memory at different scales. So DNA is much more sort of long-living medium than DNA methylation, and so on and so forth. So it's or general adaptations, I don't know, mechanics with specific mechanisms having different characteristic times. Connie, I realize that we're getting close to the cutoff time.
Till the end, yeah. So I think we've now combined the last two sections where we're getting the consensus, and then I'm supposed to talk about it. I think we've now combined that into two. I did have a thought about something else you could add in this, so this idea of memory and persistence of a DNA methylation or epigenetic signal, not only over time, but across generations, and that we would probably expect to see some variation there, that DNA methylation or epigenetic changes to certain genes might persist across generations for different numbers of generations, either as an adaptive or maladaptive response, maybe based on what the gene does, that some genes can tolerate more changes in DNA methylation or longer persistence in DNA methylation, but that in addition to thinking about the time scale and the taxonomic scale, think generation wise, and just think about that we would expect to have some variation across all of these things, not just level of methylation, but persistence of methylation across time in one generation and then across numbers of generations. I don't know.
Yeah,
made things more confusing or not with that.
Right. No, I think it's a like I don't know if I captured this by saying by mentioning sort of memory and persistence across different scales, all the way from an individual and across generations, all the way to the sort of evolutionary scale. But yeah, I think that it's kind of a another way of putting things in sort of a general context is to refer to evolution, maybe mentioning that these mechanisms can, sort of epigenetic mechanisms can play a role, not just in adaptation, sort of in a short time, but also in sort of ability of individuals to, or ability of species to evolve at different sort of speeds and scales. And that's going on from the one of my favorite papers from Susan Lindquist about Arabidopsis, sort of changing in response to heat stress, to all the way to what Eric Selker had been looking at, sort of extremely conservative policing of the genome in fungi. So Connie, you do you want to wrap up?
Yeah. So I think we have to wrap up here. It is, 2:00. Is there anything people would want to add to, what Sasha just had up on the screen about our, take-home messages, bottom line of the current state of at least a subset of funded URL epigenetics projects? Maybe you did a great job of summing it all up, Sasha.
Okay. I really enjoyed this workshop, so thank you, everyone.
Yes, thanks to everybody for, participating today, for all the PIs, for answering the 2 series of questions, that you were set. We really use that information to guide the discussion, and I think it's been really useful. Holly, did you have a hand up or no?